We almost had to swim down here, so we apologize for the delay in getting things set up. We got an early start, but we're delayed along the way with a tremendous rainstorm. So I'll now switch over and we'll continue with the lecture on the gametogenesis. And I'm in the process of getting the other lectures taped, and we'll have those posted as streaming video, and we'll get things caught up. Marco, I think you've got your podium microphone on. You might need to mute that, please. Okay, thank you. Set the pointer here. So we are now beginning the discussion of the gametogenesis for the female. We had completed the discussion of spermatogenesis for the male. So oogenesis is the term for gametogenesis in the female, and as we had stated, it begins during the embryonic period of the female, and it is completed after puberty. So it continues until menopause, and it may never be completed in some cases for individual eggs or ova. So the terminology then, oogonia, is the term for the diploid cells that undergo the mitotic divisions. So this will occur at a different stage than we had discussed in the males. It will occur during the embryonic and fetal stages, but there will be no oogonia in the newborn or any later stage of life in the female. At birth, they are primary oocytes, and we'll get into that detail and reemphasize that as we proceed. So the primary oocytes are diploid cells. They are the daughter cells of the oogonia. So in these cases, the oogonia undergo tremendous mitotic division very early and before birth, and eventually all the oogonia are lost in their efforts to produce large numbers of primary oocytes. So they will undergo the meiosis I, the reduction division, and they're surrounded by supportive cells called follicular cells, and these will be the primary follicles. So the primary follicles contain the primary oocytes, and we'll look at the histology of this in just a moment. So this becomes more complex as they are recruited into development in during the menstrual cycle, and they will become secondary follicles, as we will see. So the primary oocyte is suspended at prophase I of meiosis I cell division. And it completes all of meiosis I shortly after ovulation, and at ovulation it will begin meiosis II up to the point of the metaphase. The division of the cytoplasm is unequal, and it will yield one haploid cell and one non-functional polar body, which typically will degenerate, or it may go on to divide. The secondary oocyte is a functional haploid cell, which will undergo meiosis II, and this will produce the mature haploid ovum that can be fertilized. The ovulated secondary oocyte is surrounded by the follicular cells and the eggshell of sorts, the zona pellucida. And the cell that now has completed both meiotic divisions is a fertilized ovum, or it is an ovum available for fertilization, and there will be 3 residual polar bodies. So we recall that the original primordial cell, the oogonia, or spermatogonia in the male, will go through full meiotic divisions, meiosis I and meiosis II, or the daughter cells of those will go through full meiosis I and meiosis II to produce 4 haploid cells. So there's 3 residual bodies, 3 of the 4 haploid cells, and then the mature ovum that is capable of being fertilized, the 4th. So this slide shows us the life of a human egg or ovum. Starts with the primordial germ cell in the early embryo, and there's a migration to a zone called the genital ridge that will ultimately be the posterior abdominal wall, located in the posterior abdominal wall, and this is in the very early development. When we talk about organogenesis and organ system genesis, we'll revisit this as well for the reproductive system. So these are the primordial cells for the oogonia, and during embryogenesis, they will develop into primary oocytes and be arrested at phase 1. And the ovulation will not occur until puberty. So this arrested state in prophase 1 is from the embryonic period through the fetal period, from birth all the way to puberty when menses begins. It's only then when a menstrual cycle is initiated that some of these primary oocytes will be recruited to undergo development and develop into secondary oocytes and then become arrested at metaphase 2. And then at fertilization, the single ovum that is mature and ovulated will undergo the final meiotic division and produce the mature ovum that is fertilized. And then the female pronucleus and the male pronucleus from the sperm will fuse together, undergoing this process of pronuclear fusion to completely form the zygote. So we now begin to introduce the histology of this. So at birth, there will be primary follicles. They don't migrate around in a circle like this within the ovary. This is a horizontal section through a human ovary. And we see the neurovascular bundle coming into the core of it within the stroma. And then the parenchymal cells, of course, are the primary follicles, and they're the cells that are associated with those, as we'll see when we proceed through the next set of slides. So we see the primary follicles will be recruited, and illustratively we follow them in around the perimeter of this ovary, but indeed they are randomly selected and develop from a random population within the ovary. So the cells surrounding the primary oocyte will begin to develop, and thereby will develop into a secondary follicle, and the first meiotic division, as we said, will proceed, and this will produce the secondary oocyte up until the point where there is a mature follicle, and only a single follicle will be recruited to the point of ovulation, and then, of course, after ovulation, it develops into the corpus luteum. So we're introducing this anatomy now, and we'll revisit it when we talk about the menstrual cycle. So the meiotic events that are occurring are shown on the left, and then the histological appearance as illustration is shown on the right. So the oogonium is present. It is a stem cell before birth, undergoes mitosis to produce the diploid primary oocytes, and we see them contained within primary follicles surrounded by follicle cells, and then the growth will continue up to the point of childhood, so that there is a modest amount of growth in these cells, and they still are the primary oocytes arrested in prophase 1 that are present at birth. And then at puberty through menopause, each month one of these will be recruited into the full maturation to form the mature ovum that is ovulated and available for fertilization. So many are recruited, and we'll reemphasize this when we talk about the menstrual cycle. But right now we're following the single one that is recruited that goes through the lineage to divide. Meiosis 1 completed by one primary oocyte each month. Then a secondary oocyte is arrested in the metaphase 2, forming the secondary oocytes. And then at ovulation and fertilization, the final division occurs. And we see the polar body, the first polar body is formed. It will undergo the final meiotic division along with the final meiotic division of the larger secondary oocyte. The secondary oocyte, when it undergoes the final meiotic division at fertilization, produces the second polar body. So we end up with 4 haploid cells, only one of them capable of fertilization. And we'll see this illustration of the developing follicle to the mature vesicular or Graafian follicle, that single one that reaches this point out of the group that is recruited each cycle, and it will undergo ovulation. So recall the 2 distinctions between the male and female gametogenesis. The male gametogenesis begins at puberty, so that's when the spermatogonia begin to divide and produce primary spermatocytes, and spermatogenesis can occur throughout the remainder of the life of the individual. However, in the female, the division of the oogonia to form the primary oocytes is in the embryonic and fetal stages, and the final meiotic division, the final developments, don't begin to occur until menses and terminates at menopause. And there is an ongoing recruitment of these. So here we see 2 histological samples. On the left we see primary follicles, and on the right we see one of the follicles that has begun to develop. So first on the left we see the large cells here delineated by the cytoplasmic membrane, and we can see the large nucleus within it. On the right-hand side we see a follicle with arrows in it, but we are at a plane of section where the nucleus is not visible. So if you think of a basketball with a baseball in it, when you make cuts through the basketball, not every slice through the basketball is going to be through the baseball also. So this is a key aspect of histology. Over here on the left, we see a profile that we have just cytoplasm and not the nucleus of a primary follicle. So take note of the arrangement of the follicular cells surrounding the primary follicles. In some cases, they're more flattened cells, and you don't see the nucleus lined up all the way around. And then some that are at another stage of development will have very cuboidal type of cells. We see the nuclei of all these cells. So these are little cubes of cells all lined up surrounding this primary follicle. So this one has been recruited to undergo development. These very likely are sitting and waiting for their turn at some later cycle. This follicle on the right is at a later stage of development. So this layer of cells, follicular cells, has undergone mitosis by stimulation due to FSH from the pituitary gland, as we will mention. So the follicle stimulating hormone is stimulating recruitment and development of the follicles. So there's mitotic division of these cells to increase in number and their synthetic capacities, as we will see. This green arrow is pointing to a thick structure surrounding the cytoplasmic membrane of the oocyte, the eggshell, if you will, and this is the zona pellucida. The blue arrow is pointing to some of the connective tissue stroma cells that are beginning to differentiate into a theca or a tunic that is surrounding the the oocyte. So it's called the tunica interna or theca interna, as we will see. So these cells, the follicular cells and the theca cells, are going to interact to produce the female gonadal steroids. In this view, we are seeing a secondary oocyte that has begun to develop the antrum. So this is one that was selected to go on to become a mature follicle. Again, the green arrow is showing the zona pellucida, and we see many of the follicular cells, mostly the nuclei is what we see, directly surrounding and sticking to the secondary oocyte, and the fluid-filled antrum is beginning to appear, and many of the follicular cells stay distributed around the periphery of the follicle, and we can see the tunica or the theca is becoming more and more developed, so there will be a theca interna and a theca externa that will develop in these follicles, and those are shown here. So the green arrow is showing the theca externa or tunica externa, and the blue arrow is showing the theca or tunica interna. So these are very important cells. These are targeted by LH, luteinizing hormone, and these follicular cells are targeted by FSH, and collectively you get follicular growth and development and the production of estrogen. This is a summary slide showing the primary oogonia undergoing mitosis, so proliferation to produce more oogonia that undergo growth, mitotic division to produce more oogonia that undergo growth and differentiation to become primary oocytes. So this is a bit different than what we had mentioned for the primary spermatogonia. There still was growth and proliferation very early on in the development of the spermatic cords in the immature testes, so that there is an infiltration of many and numerous primary spermatogonia, and then they are arrested at that state until puberty. In the female, there's proliferation to produce many oogonia, and then finally just growth and development with further division to form primary oocytes, and that is what is present at birth. And then the oogenesis is the sequence of events from the primary oocyte that we described to the mature ovum that is available for fertilization. This slide shows us a low magnification of human ovary, and we see many populations of follicles at different stages of development, and one that is reaching maturity. So there's a single in each menstrual cycle. There's a single ovum. No one really knows for sure whether it's right or left ovary, whether it alternates, the same one every time. But there is typically and most commonly a single follicle that is recruited to the full mature development. This one still hasn't reached the edge to bulge out and be ready for the rupture outside of the ovary, ovulation in other words. But we can see the levels of recruitment and development of the primary follicles. Some primary follicles over here have not been recruited. They might be recruited the next time. This is a series that was recruited, and we see some that are approaching secondary oocyte, secondary follicle stages, but they will not continue. They are contributing to the synthesis and secretion of estrogen and a little bit of progesterone, but eventually this follicle will take over and be responsible for that. And of course, this is the one that would ovulate and differentiate into the corpus luteum. We note on this a 1 millimeter scale bar. So this mature, nearly mature Graafian follicle is not quite 1 millimeter in diameter. So eventually the edge of that mature follicle will approach the surface epithelium of the ovary. And this is what we're seeing in this field of view here. So there is a basement membrane that separates the follicular cells from the theca cells, or the tunica cells. So the theca interna is closest to the basement membrane, theca externa is farthest away. And eventually this will bulge on the surface of the ovary, and there will be a variety of secretion of local hormones, mostly prostaglandins, that will activate enzymes as ovulation approaches, and the connective tissue here will be broken down such that the ovum can be ejected from the antrum. All the time this is occurring, we get the growth in the size of the follicle, and it's approaching the edge of the ovum, the ovary. The primary oocyte is sitting on this little clump of follicular cells called the cumulus oophoris, shown here highlighted in the orange color. We see the zona pellucida in blue. And then in the upper right-hand side, we see the ovulated secondary oocyte, and it is surrounded by some of these follicular cells that accompanied it, and now these follicular cells that accompanied it at ovulation are called the corona radiata. So it's a radiating crown of cells that surrounds the oocyte. So here we see the corona radiata of some of those follicular cells that accompanied the ovulated ovum. And it is now undergoing the final division, meiotic division. And we see it is at the same time that sperm are contacting the corona radiata, and the acrosome is broken open, and there's enzymes that degrade the connections between the corona radiata cells and eventually the zona pellucida. There's a tremendous amount of signaling by the ovum to attract the sperm and to control actually the access of the sperm. The ovum plays a big part in selecting which sperm will actually penetrate the deepest and be able to degrade through the zona pellucida, and then there will be a final reaction once there's contact between the sperm and the cytoplasmic membrane of the ovum that we'll talk about in a bit more detail later. So indeed, in panel B here, we see that one sperm will be allowed to fertilize, and the nucleus from that sperm will enter, so that there will be the pronuclei shown here in panel D. They will come together, and the paired chromosomes will, the chromosomes for the appropriate pairs will pair up and undergo unification. I went from C to E. C to D, the pronuclear membranes will break down, and then we get the alignment of the chromosomes to be paired, and they will pair together, and then, of course, begin DNA replication for division. So here we have a slide with a question on it. Immediately we should recognize this structure as a primary follicle with the zona pellucida, a primary oocyte with the zona pellucida and the follicular cells and the developing theca interna shown here. So Q1, this structure is a primary oocyte within a primary follicle. And then Q2, what cell type is found here? Of course, these are the granulosa cells. So these follicular cells that we've been referring to, we're now giving a more specific name called the granulosa cells. So the follicular granulosa cells are the ones that immediately surround the developing oocyte. So the highlighted structure here, it's highlighted in pink in this follicle. In this follicle, we know that it's the zona pellucida. So it appears between what two structures? So these are the granulosa cells, and then this is the oocyte. And here So it would be between the oocyte and the granulosa layer of the follicle. The highlighted structure here, so this is the red highlighted structure, is the it is the cumulus oophoris, and of course the cells originate from the granulosa layer of this structure. So it's not the theca, not the zona pellucida, nor from the oocyte. It's from that granulosa layer of those follicular granulosa cells. So you can see that there's going to be quite a few histology slides for you to answer questions on the exam. And there will also be some where you'll have to label structures as we see in this slide. So structure number 1 should be the theca externa. Number 2 would be the theca interna. Number 3, so you can see that these are listed in the same order as they are numbered here. So number 3 is the follicular antrum that has fluid. Number 4 is the beginning of the cumulus oophoris. Number 5 is cells that will eventually be part of the corona radiata. Number 6 is the membrana granulosa, or the granulosa cells that are now beginning to form a lining of the antrum, so it's referred to as a membrane of cells. Number 7 is the oocyte itself. Number 8 is the zona pellucida surrounding it. And then number 9 is a space called the spacum perivitilinum. Spacium perivitilinum. A little bit of Latin for us there, I guess. So whatever you have there, you can find a name for it. And you should be able to label all of the structures that you saw indicated on that slide. So in humans, each diploid oocyte gives rise to 1 ovum and 3 polar bodies. So we said that we'll always have 4 haploid cells as the final outcome of meiosis, meiotic division, of gametogenesis. And in the female, the primary oocyte will ultimately produce 1 mature ovum and 3 polar bodies. And then all of the other follicles that are recruited do not complete meiosis. Eventually, those follicles and the ovum contained, the oocyte contained within them will undergo degradation. Actually, it's a type of cell death referred to as apoptosis. A-P-O-P-T-O-S-I-S, apoptosis. And that overall process of the loss of the follicles that do not mature into a Graafian follicle is called atresia. So those follicles that are undergoing the apoptotic cell death are called or referred to as atretic follicles, A-T-R-E-T-I-C, atretic follicles, follicles undergoing the process of atresia. So occasionally 3 polar bodies are found clinging to the mature ovum. 1 came from an unequal division of the ovum, but from where did the other 2 arise? So we already know from the previous illustrations that it's the first polar body that divides to produce those, and let's see where that is. The first polar body has also divided to produce 2 polar bodies. Oh, that's not really. Yeah, that is correct. So I'm moving, but my pointer isn't. So for some reason, the computer. Oh, there we go. Must have been one of those things trying to get in, huh? Did you guys set the security on this? I know you can see the question. I just can't move my pointer. There we go. So each time a human germ cell gives rise to an egg or sperm, a total of blank combinations of maternal and paternal chromosomes are possible. Holy crap, I forgot the answer to that question. So each time a human germ cell gives rise to an egg or sperm, a total of how many combinations of maternal and paternal chromosomes are possible? I'll have to look that one up. Unequal cytoplasmic divisions are characteristic of the oogenesis. So in spermatogenesis, it's equal division of the cytoplasm. In prokaryotic fission, typically it will be equal. In mitosis, there is an equal division of the cytoplasm. So it's only in the oogenesis with the formation of the polar bodies that there will be unequal cytoplasmic divisions. So that gets us close enough to the end of class time. I will begin the next lecture on Friday, and we will step up the pace a bit through the in-class lecture material, and we'll get on track. The lectures that need to be taped will get taped this week and early next week, and Robert will post those as streaming video for us. So I appreciate your time and attention, and especially your patience in waiting for us to deal with the technical challenges. Are there any questions? We have a few minutes. No questions? Nobody wants to be on camera? Slow down because I asked him because for me. Let's see here. And this booth without requiring a login now too.
Yeah, they did that same thing over here to that one. I did that also, and it did work out. Let's see remote networks. These are just to connect to all these rooms. Well, he connected to you from that room over there. That's why your computer froze up for a second.
Okay.
Because we're trying to see we could send over the network.
Will this connect to the computers in here?
Yeah, this will connect to the computers in here, and we can connect from over there back. That's what he did. He accidentally took over your computer for a second, so he was moving the mouse for a little bit. But we're trying to do it. See if we can do it back the other way, but it doesn't look like it has it on here. Well, here it says APL. Let's see.
So then, if somebody was going to teach a computer science class,